to such an extent that when the Human Genome Project was completed, they felt having a haploid copy, just what the genes were on one copy of the chromosomes, was enough to answer our DNA, whatever that means. Okay, so it was really quite a novel concept. So once that became apparent in 2004, 2006, they went back to, I don't know if you've heard of the HapMap populations, but that's a group of 270 people from 4 different ethnic groups, Northern European, Han Chinese, Japanese, and Yoruba, that had been used to figure out the variation in SNPs, in these single nucleotide polymorphisms. And they said, oh my God, we didn't even know we had this kind of variation. Let's go back and look at them. So they looked at those people. They screened them for copy number variants by 2 different methods and found 1,447 copy number variable regions covering 360 million base pairs of DNA. 12% of the genome is copy number variable in just those 270 people. Okay? And that variation is more variation than we know about in SNPs, in which we have about 4 million from person to person. Okay? I don't know if your mind is blown, but mine is. Okay, so now where is this? It's everywhere. Okay? Sometimes it's associated with little repeat areas, and that predisposes to not aligning correctly, but sometimes it's not, and it's everywhere in the genome. Usually in small numbers, but sometimes very frequently. Okay. Recently, this is last October, the 1,000 Genomes Project finished looking at 1,000 genomes, and looking for copy number variation. They found 20,000 different structural variants that most had not been previously reported. Okay, so this is an enormous area of variability between us. You know, we're all supposed to be the same. Well, this is an area where we're not all the same. Now, this is an output of a SNP genotyping platform, and what you can see is, I'm going to step down here. This is normal. This is 2 copies. Right here we went up, and there's 3 copies. Same thing here, we have 2 copies, and here we went down, there's only 1 copy. When you do it on a SNP platform, not only do you get the intensity read, you also get a genotype call. So this is homozygous for one allele, homozygous for the other allele. This is heterozygous. Where you're missing a piece, you can't have any heterozygosity, right? Because there's only 1 copy. So this is a nice double check. Same thing here, you go along and you have an extra, and what happens is you don't have heterozygosity, you've got either 2 of the same and an extra, or 2 of the other same and an extra. All right, it's called AAB or ABB calls. So this is a very nice way of seeing what this looks like. And the other thing is it's kind of machine readable. This does not need a human being with 20 years experience to make sure they get it right. Okay, so this method allows us to detect all kinds of new conditions that were previously completely undetectable. So this is a child who has a 17q21.3 microdeletion, previously undetectable. It's only 600,000 base pairs. There's no way, no microscope is going to ever be able to pick this up. And now what people are doing is they're looking across the entirety of the genome to see where there's missing and extra pieces in people with problems. But we also have to know about the missing and extra pieces in people who don't have problems to figure out if it might be causative. Now, why would an extra or a missing copy of a gene be a problem? First of all, there's some genes for which 2 copies is absolutely essential. 1 is not enough, and 3 is too much. Also, where these breaks happen might actually disrupt the gene, a gene that's very important to have 2 copies of. So it might not be actually what's in the interval, but right at the point that the break occurs. And also, even if it doesn't, you're not missing a dose-sensitive gene, it might be a regulation, an area that regulates genes either upstream or downstream of where that missing or extra piece is. Okay, so how do we look at this? A number of different ways we can look at this. Back arrays are almost passé, and this is not that old a slide. Oligo arrays look only at dosage, and almost everybody's using 180,000 probes. Okay? SNP array will look at dosage and also the genotype call that I just showed you a second ago. It gives you the 2 pieces of information. Will also tell you if people are related, so it gives you additional information you didn't have from just looking at intensity reads. And some of the labs are also using another panel made by Affymetrix. And LabCorp, which is one of the largest labs in the country, will call anything less than 200 kb. They just give you a report of a normal result. Now, we all know we have CNVs, but they don't tell you what they have. They just tell you normal. Okay? Unless it's more than 200,000 base pairs, then they'll tell you, send me the parents. Now, by doing this whole genome approach, you get even more things. So this is a rearrangement at 1q21.1 that has such variation that actually you can't tell anything ahead of time to know. There might be something wrong with this kid, so you send this test. And you find this abnormality. So what they're saying is actually, you now make a diagnosis based on genotype, not on phenotype. So before you came in, clearly there's these things wrong with you. I'm supposed to figure out a syndrome. Now, there's something wrong with you. Let me send this unbiased search of the whole genome and see what's missing or extra. It's a very different approach to practicing medicine, and it's what everybody's doing now, standard of care. What happens with this method, though, and you can imagine it's brand new, right? We didn't know this existed till 2004. We only had the HapMap people in 2007. What we find is things that we don't understand. So there's more and more normal people being done, and there's more and more abnormal being done, and eventually those variants of unknown significance will sort to normal or benign or pathogenic. But at the moment, we get a lot of things that we don't understand. Matter of fact, up to 20% of the results that we have are variants of unknown significance, which leave us standing there going, well, I don't know, but keep coming back to me and I'll keep reading and maybe we'll be able to sort this out. But that's the kind of medicine I have to practice now, a lot of variants of unknown significance. And when we get these, we look at how big is it? Is it inherited? So the presumption is if it's inherited, it's less likely to be pathogenic. However, if the parents aren't normal, you really have to rethink that conclusion. And then the last thing is that you ask, are there disease genes in the interval that are known to be dosage sensitive? Because then it's more likely to be a problem. And you go to a database called OMIM, and here's where I have to disclose that I run this database, so I'm not unbiased. Okay? And this is OMIM's homepage. Have any of you heard of OMIM before? Got a couple yeses. Well, you better say yes. So we have a new website. It's omim.org. Okay? And that's what the homepage looks like. If you look at statistics, this is just for your information. There are 20,559 entries in OMIM, which catalog about 13,700 genes. So we have more than half the genes cataloged in OMIM, but not all of them. We focus on genes that are known to be associated with disease and the genes of known function. We don't worry about genes that nobody knows what they do yet, but we'll get them in when we can. And then the other thing is, people always ask this question, so I put it across the ticker at the bottom. There are currently mutations in 2,565 genes causing 4,321 disorders. This is a number everybody wants. You got it today. Okay. And it will be showing up on the statistics page of oming.org on a daily basis very soon. We're working on programming it. So here I put in 1Q21 deletion. That's the one that I told you that you don't know what you're looking for. You just look for it. So we have it. You can go, that if you search for that, you'll get this read. If you click on it, I wanted to decrease my slides so you can again find out all about it, how the patients are described, what they have. Now I want to tell you that CNVs, de novo copy number variable regions, seem to be very important in neuropsychiatric disease, in schizophrenia, in autism, in seizures, in mental retardation. And people who would like things to be clean are very annoyed by this because there are all kinds of copy number variations, and the same ones seem to predispose to all of them in different circumstances. So Ada's simplistic view of this is that we have 22,500 genes, and we have an environment, and the brain is very, very, very complex, and brain genes are strewn throughout the genome. There's no particular chromosome they like more than another. And so if you disrupt anything, you are likely to disrupt a brain gene. And then what phenotype you get, if any, depends on all the other genes and the environment. Which is why I'm not at all surprised by the fact that all of them predispose to the same things and different things in each person in a family. That doesn't bother me at all. Bothers other people.